Hello, my name is Kennedy Nguyen. I'm the lab manager for the imaging and microscopy facility at UC Merced. My job is to train students and faculty and staff for using the equipment at the imaging facility, such as confocal microscopes, electron microscopes, and different instrumentation to assist in materials characterization. As you can see, this is a wall of the different grants that different faculty on campus has used to provide instrumentation for our lab. So I hope you enjoyed this virtual tour. Hello, this is our first room. This is our sample preparation room. Since most of the equipment we use in here is for electron microscopy, we need to make sure the sample is conductive, grounded, and can survive in a vacuum. A lot of materials don't fit that criteria, like for plants aren't the most conductive material. So this instrument here is our critical point dryer. What we could use, if we have a plant sample, we could critically dry the sample, make sure it can remove all the water moisture so then it can survive better in vacuum. And then we could use one of our other instruments. We could coat the sample after it's completely dry, and then we could analyze it with electron microscopy. This instrument here is our plasma coater. To add materials onto the different stubs or different grid materials, we need to make hydrophilic or hydrophobic. So this is how we treat our samples with different gases, oxygen, hydrogen, argon, to treat the sample surface so we can analyze the sample as well. This machine over here is our carbon coater. If the sample is not conductive, then we could add a thin layer of carbon coating on top of it to make it more conductive for electron microscopy. This instrument over here, inside the fume hood, this is our Vitrobot machine. This is for freezing materials into a drop of water, so then we could view it in transmission electron microscopy. This is our Zeiss Gemini SEM 500. These are a couple samples we have here. We're going to use on the microscope. We have some plant samples. We critically point dried them, and then we coated them with gold. And now we're going to look at put it in the SEM and have a look at it. Okay. Okay, so we already have our sample inside the sample chamber here. Earlier we had to vent the chamber since it's always in vacuum, put the sample in and then pump the chamber. So now that it's already pumped, we have stage controls here moving up the sample. You can see our sample here. And we're moving it close enough so we get a higher resolution imaging depending on the working distance here. So first we'll go to low mag. Check out zoomed out to see, we'll orient ourselves on the sample. All right, so let me get the zoom here. So we have our sample here. We can move around, see if there's any interesting parts on this leaf here that we're interested in. Here's like a dirt particle or something because you see it's charging. Usually the black and white usually indicates some kind of charging. Okay, so we have that. I'll save it. And then our images will appear over here. So you can keep track. This is our picture of our sample on the stage. And then we have a picture of the leaf here. Okay, so this instrument here is our Quanta 200. It's from FEI, or now Thermo Fisher. This is our environmental scanning electron microscope. It also is equipped with an EDX detector, which is energy dispersive X-ray analysis. We're when we hit our samples with electrons, not only do electrons bounce off the sample, but there also produces X-rays. And these X-rays will be characteristic of what kind of material that the electrons are hitting. So analyzing the X-ray signal from the electron microscope signal, we could analyze what kind of element the material is. So here we have our sample inside. KV energy is going up right here. Let's auto brightness and contrast of this. Okay, there you go. Now we're seeing stuff. So let's here. Let's increase the pacing. So I'm gonna focus, making sure we're seeing something. We have our sample. So this is a map I did before. So this is a view of the sample, and then these are the different spatially where each of the elements are. So there's light, so you don't see a lot of carbon, so you don't see the signal. You see oxygen. This is where the oxygen is located. Magnesium, and you can see aluminum, silicon. Some of these there's not as much signal from these elements, but they're there. For the calcium, you'll see a lot of shadows right here. X-rays travel in straight lines. So if the X-ray doesn't, this is where the sample is. The X-ray detector is about facing us here. So when the electron beam hits the sample, all the X-rays have to go straight directly to the detector. But if the electron goes into a hole and the X-rays are formed, if it hits the sample, then it won't go to the detector. So then you'll see shadows inside the sample. So that's why the image of the sample, see all the holes here. And in these maps here for the elements, we can see the holes representing some of the electrons or X-rays that didn't make it to the detector. Only the ones on the surface did. This is our X-ray diffractometer. This is used to analyze the crystal structure of different materials. We can analyze solid materials. Here, I have a silicon wafer on this disc holder here. Or we can also analyze powdered samples. So if you have a powder of silica powder, nanoparticles, you can put the powder here, flatten the surface, and then put it into the machine to analyze the crystalline structure of the material. So I'm going to put this into the machine, and we're going to see what kind of crystal structure it has. So we open the doors, add the sample slot. Okay. Close the doors. So what's going on here? So the X-ray is going to hit the sample, and then we have a detector that's on the side. X-rays are always going to be scattering in different directions, but the detector is going to detect at which angle the X-rays are going to bounce off of. And at every angle, it's going to collect the angle. As you can see, the arms are moving. And then we'll get a corresponding spectrum, 35, 55. At these different angles, we see diffraction occurring. So this is basically the fingerprint of the crystal structure of the material, or the silicon wafer that I put inside the chamber there. This is our transmission electron microscope. It's a little different from the two scanning electron microscopes that you guys saw earlier. The scanning microscopes scan on the surface of your sample. For this one, the transmission electron microscope needs the electron beam to penetrate through the sample, so then we'll see the sample. So the kind of size that we need is about a 3 millimeter disc. So you see these grids here. This is about the approximate size you'll have for the holder. So I'm going to take out this holder so you can see how thin and how small we have to put on the grids, so then we can put it into the microscope. To put the grid on this sample, we use this tool here. Insert, lift up sample here. And then we would grab one of these tweezers here. Then if I would grab I'd grab one of the TEM grids, and you could see I'll place it onto here, center it. Once I do that, place this back. Then normally when we put the TEM grid, we gotta check, make sure it doesn't fall off. We'll turn it 180. So there. But let me show you the other types of holders we have as well. This is our single tilt holder. This is our double tilt holder. Just like the name suggests, so when we put these samples into the microscope with a single tilt holder, we're able to tilt it one direction. For the double tilt holder, we can tilt it in this direction. We could also tilt it in this direction. So if we're looking at a crystalline structure, we can look at it at different angles. Okay. We also have this is our cryo holder. Okay. It's being pumped down right now for a cryo experiment later on, but yeah, so that's being warmed for when we need to use our sample. So I'm going to align the small pin to this arrow key here. Have the pump start, single tilt selected. You can see we can use different holders, the cryo holder, the double tilt holder. We use a single tilt here. Select. We'll just have this pump for 2 minutes. So this is the interface for the TEM software. So once we have the sample inserted, we'll open the column valves to let the electron beam go through. So you can see this is our sample here. I'm going to zoom out so you can see. Go to lower mag. Okay. So this control here, I'm controlling the spread of the illumination of the electron beam spreading out over the sample. So you can see this is typically on the TEM grid, you have these meshes. This is a copper mesh, and then you have your sample grid or your sample in between this layer here. And then we could zoom in any of these sections here to see what kind of sample we have here. This is a wafer sample. So let's see here. Just zoom in. I'll condense the light as well. Or in this case, condense the electrons. So right here, what I'm doing here, so we could also operate the TEM like a scanning electron microscope. And this mode is called STEM, Scanning Transmission Electron Microscopy. So it's gonna do a scan of the image here. So instead of like the image we saw earlier, this is a scan of the surface. So we could take a picture here if we wanted to. So you can see it's scanning across line by line, and it takes an image of the sample there. This software over here is for analyzing the chemical composition. So after the scan is done, I'm gonna import the image to the software here, and then I'll analyze the chemical composition of the sample here. So we're sending the image over. Okay. So this is the bright field image from the transmitted electrons, and these are just an image from the diffracted electrons here. We have a foil of aluminum foil, and then I'm pinning them down using these clips that are loaded as springs. We're trying to keep the sample flat, and I have it on this platform to make sure the height isn't too big. So this is our height checker. Making sure the sample isn't too big. If it was too big, we could, as you can see these knots here, we could adjust the lock. We'll move it up and down, and we'll move the stage up and down if we needed more height for the sample. So the sample height could range between 0 to 20 millimeters. Okay. So I have the sample height checked. So I have the sample ready. So then I'll place it into the holder here. We have these knots here, they align to these prongs there. So I'll align this here. Make sure it's solid. Okay. Then I'll close the door and then start the pumping process to the analysis check. See how it's focusing for me? It's doing auto height, getting the optimal height at this sample here. So you can see the focus is getting better once it finds the optimal the height for the sample. And then starting the scan for the first sample, so I'll go back to data view. You get our spectrum. It's going to do 2 scans. So this is our second scan. And the next 3 scans are for aluminum, carbon, and oxygen, or what I suspect should be there. Since it's aluminum foil, it should have aluminum. So this is the scan for aluminum. And the last peak is for oxygen. Okay, so there's oxygen as well. All right, and then if you didn't, so let's see.
So we'll analyze the data here. Since I knew it was aluminum, so I was able to, I chose aluminum, carbon, and oxygen. But if, say, you didn't know what the sample was, you could also do an XPS survey, analyze the whole spectrum, and then do an auto ID and have the software analyze your sample for you. So you can see here the software automatically identified the peaks for aluminum, oxygen, and carbon. So for XPS, this is really good for analyzing the surface of your material and determining what kind of materials are present on the surface. We have when you characterize different materials, the bulk or what's inside the sample may be different from the surface. So especially when you're looking at nanoparticles, the surface properties may be a lot different from the bulk properties for the whole material. So XPS is pretty useful for analyzing the surface properties. This is our confocal microscope. This is for imaging samples with fluorescence. Sample in there, I'm adjusting the illumination so I could focus. Sample there. All right, so once I have the illumination set, okay. So I already have I turned on the different lasers on earlier. So now we're gonna check if there's any fluorescence on the sample using the different lasers. What I'm gonna use is, let's open. I'll try my area scan. All right. Okay. Let's stop there. Okay. So some of it's a little too bright, so I'll adjust that. Okay. So this is the signal here. Let's set the exposure. Okay. So it's not as intense. Okay. So let's set the exposure. So you can see the signal from the red. And then I'll set the exposure for the green as well. Okay, so you can see each of the pictures look a lot better. Okay, so I set the exposure for every single signal. So now I could let's take a picture of all the signals together. Snap here. So the red, the green, and what's fluorescing blue. So you could locate, based on which whatever's fluorescing, you could ID whatever material. So let's say if you had nanoparticles, and you could have them attached to specific parts of the tissue and identify where they are. So you can see the green is located here, red is located here, and you all basically quantify whoever is fluorescing with what it may be potentially based on the fluorescence there. Okay.
And that concludes our tour. I hope you enjoyed our virtual tour of the imaging and microscopy.